Hello, and thanks for joining us today for the latest Technology Networks webinar, Strategies for Isolation of Plasma Membrane Proteins. I'm Karen, Science Writer for Technology Networks, and I'm here to moderate the event. I'm very pleased to have Betsy Benton and Joanna Geddes joining us today as your presenters. Betsy completed her PhD and postdoctoral research at the University of Wisconsin-Madison in the fields of biochemistry and human oncology. She joined Thermo Fisher Scientific in 2000, where she's developed numerous products in the areas of protein sample preparation, including membrane protein isolation and cell fractionation, protein purification with both magnetic beads and resin supports, and protein detection. Joanna is an R&D scientist at Thermo Fisher Scientific, where she has actively worked on the development, commercialization, and support of protein sample preparation and protein interaction products introduced over the past 10 years. Joanna has experience in a multitude of techniques such as MS sample prep, cell and tissue lysis, cell separation, and protein expression and extraction. She's worked on the development of products for dialysis, protein concentration, membrane protein extraction, protease and phosphatase inhibition, and immunoprecipitation. She received her BS in microbiology from Western Illinois University. A very warm welcome to both of you. Following the webinar, we will have a Q&A session. We would welcome any questions you may have. Please ask questions by entering them in the text box on the left-hand side of your screen and pressing send. The Q&A session is a great opportunity to gain valuable insight from Betsy and Joanna, so I very much encourage you all to ask questions. Any questions we don't have time to get to, we will be sure that you are contacted offline with an answer. If you experience any technical issues during the presentation, please click the question mark in the top right-hand corner to request support. Please remember you can ask questions at any time, even if you're watching this webinar on demand. I'll now hand over to Betsy and Joanna.
Hello, my name is Joanna Geddes, and in today's presentation, we will be discussing the isolation and enrichment of plasma membrane proteins. I will be going over an introduction where I'll touch on the challenges scientists face, as well as the importance of plasma membrane proteins. Then I will dive into the techniques for solubilizing functional G-protein coupled receptors from the cell membrane. After that, I'll hand off to Dr. Betsy Benton, who will go over the techniques for the enrichment and isolation of cell surface proteins. And at the end of our talk, we'll hold a question and answer session. Plasma membrane proteins act as the gateway to the cell and are involved in many critical cellular functions. In this sample prep workflow, we are focusing on both the extraction and enrichment of these important proteins. Isolating membrane proteins has many challenges to overcome. Their complexity makes them hard to clone and express, and because they are embedded in the plasma membrane, extraction can be difficult and often requires harsh conditions that result in denatured or unstable proteins at low yields. This then requires an enrichment of your target. Today we will show you ways to easily extract functional GPCRs that maintain that function over time. We'll also show you an optimized protocol that not only enriches for cell surface proteins, but also greatly reduces contamination of intercellular proteins in your sample. While difficult, extraction of membrane proteins is important to understand the specifics of what's happening inside of our cells. They are responsible for the maintenance of many cellular functions, such as signal transduction, cell integrity, intracellular and extracellular transport of molecular solutes, and cell-to-cell adhesion. Peripheral membrane proteins are temporarily attached to either the lipid bilayer or to integral proteins and dissociate following a treatment with a polar reagent. Integral monotopic proteins are permanently attached to the cell membrane without passing through it. They are typically anchored into the lipid bilayer by a fatty acid, isoprenyl, or GPI anchor. Transmembrane proteins span the entire phospholipid bilayer and can be single or multi-path. They have one or more segments composed of hydrophobic residues that interact with fatty acyl groups of the membrane phospholipids. Transmembrane proteins include transporters, linkers, channels receptors, enzymes, structural membrane anchoring domains, and proteins involved in accumulation and transduction of energy, and also proteins responsible for cell adhesion. They are a major class of targets for drug development. Increased extraction efficiency of stable and functional samples is needed for the assays used for this development. G-protein coupled receptors, or GPCRs, are transmembrane proteins that span the membrane 7 times and play a key role in transmitting signal across the cell membrane. They are effective drug targets for genetic and immune system disorders and serve as potential biomarkers and therapeutic targets for cancer treatment. There are currently over 400 drugs approved by the FDA that act on over 100 unique GPCRs. As I previously stated, one of the major challenges in GPCR research is the ability to efficiently extract the receptor from the membrane without compromising integrity of that receptor. Today, we'll be looking at extracting the group of receptors on the right from multiple sample types. These GPCRs are associated with several diseases and are prevalent in today's literature. There are many strategies to extract GPCRs from both cells and tissues, and here we will touch on just a few. Subcellular fractionation uses one or more of the properties of membrane proteins, such as density, surface size, and shape, to separate based on differential centrifugation. An aqueous polymer two-phase system is a technique used that involves liquid-liquid fractionation. High salt or high pH buffers can be used to disassociate proteins from the cell membrane, but can also damage the protein's integrity. Most of these traditional protocols are laborious and time-consuming and require expensive equipment for ultracentrifugation, as well as extra steps to stabilize your target protein. For these reasons, we decided to focus on detergent extraction. Like the components of a biological membrane, detergents have hydrophobic associating properties as a result of their nonpolar tail groups. In addition, detergents are water-soluble and allow the dispersion of hydrophobic compounds into solution, such as the extraction and solubilization of membrane proteins. At higher concentrations, detergent monomers aggregate into structures called micelles. Detergents can be denaturing or non-denaturing with respect to protein structure. Denaturing detergents can be anionic, such as sodium dodecyl sulfate, or cationic, such as ethyl trimethyl ammonium bromide. These detergents totally disrupt membranes and denature proteins by breaking your protein-protein interactions. This is not ideal for the maintenance of both structure and function of GPCRs, and for that reason, we decided to focus on non-denaturing detergents for extraction. Non-denaturing detergents can be divided into non-ionic detergents, such as Multicides, Triton X100 and MP40, or zwitterionic detergents, such as chaps, as seen in this table. When choosing detergent for your extraction, it is important to take a few factors into consideration, such as the CMC and the cloud point. The critical micelle concentration, or the CMC, is the minimal detergent concentration at which micelles are formed. Because micelles are thermodynamically stable, they can provide a temporarily stable environment for your extracted protein. When the concentration is lower than the CMC, only monomers are observed. And at concentrations higher, both micelles and monomers coexist in the same solution. The cloud point, or the temperature at which the micelles aggregate and form distinct phases, is important to consider for incubation temperatures and can also be useful if performing phase separation techniques. After testing a variety of detergents, we determined that a mixture of non-ionic and zwitterionic detergents gave the best results when extracting GPCRs. Choosing the right detergent is important for solubilization, but the receptor also needs to maintain their native form and function, which cannot be done with detergent alone. As integral membrane proteins, they are generally unstable when removed from their membrane environment. Because of this, there are different strategies for stabilization, and here are some examples. Proteoliposomes are systems that mimic lipid membranes and incorporate the protein within. Nanodiscs are nanoscale planar discs of lipid bilayers stabilized by membrane scaffold proteins. SMALP is the spontaneous encapsulation of proteins from a membrane into a nanoscale styrene maleic acid lipid particle, which is where it gets its name. Amphipoles are short amphipathic polymers designed to absorb tightly onto the hydrophobic surface of the receptors and cover them with a thin layer of surfactants. And sterols stabilize by directly interacting with your GPCR that's encapsulated within a detergent micelle. Commonly, extraction is followed by a separate step for stabilization. We determined, however, that the receptor is better stabilized if both the extraction and stabilization happen in conjunction with each other. Because of this, we developed an all-in-one formulation that contains all of the components needed to extract stable and functional GPCRs. With the ease of an all-in-one formulation, we also wanted to optimize the extraction protocol seen here to be fast and efficient. You start with your sample, whether that be cells or tissue, and wash your sample in cold PBS. You spin down and discard your wash, and then the resulting pellet is homogenized in the GPCR extraction and stabilization reagent and incubated for 30 minutes at 4 degrees Celsius. Post-incubation, the cell debris is pelleted, and from there you will remove the supernatant containing your functional receptor and proceed to either further steps to purify your tagged receptor from the whole cell lysate or take your extract directly into your downstream applications. There are many downstream assays that require GPCRs to be in a functional state post-extraction, as represented here on this slide. In the following data presented, we focus on analysis of binding by immunoassays, specifically Western blot and immunoprecipitation, as well as ligand binding assays using radioligands. In this data set, we are focusing on endogenous receptors, GPR120, serotonin 1A receptor, thrombin receptor, and metabotropic glutamate receptor 3 extracted from cells in the blots on the left and tissues in the blots on the right, using two different solubilization methods. Lanes A and B are membrane preps, where the cytosolic proteins were removed before solubilizing membrane proteins with two different buffers. Lanes C and D are whole cell lysates using the GPCR extraction reagent and a ribolysis buffer. On the bottom of the blot panels, S represents the soluble fraction where you would expect to see your solubilized GPCR, and P represents the insoluble pellet or what was left behind. Focusing in on lane C, we can see that nice solid band showing solubilization using the optimized detergent extraction with our GPCR reagent. That solubility of the target is greatly improved over the detergent-free methods seen in lanes A and B. We also see better banding of the target when directly comparing the GPCR reagent to RIPA in lane D, especially in the brain and liver extracts due to less viscosity, or what I like to call goobering, in the lysate. After confirmation of endogenous expression of GPR120, a taste receptor that is linked to obesity, we took it a step further and decided to perform an IP using protein AG magnetic beads and a rabbit polyclonal antibody specific to GPR120. And looking at the Western blot results on your right, you can see the presence of the target in the flow-through lane labeled FT, and you can also see it in our elution lane labeled E, with the majority being in the low pH elution, showing a good binding of the antigen to the antibody. You can also see in the bead boil lane labeled BB that there is no GPR120 present, indicating the elution was efficient. Next, we wanted to determine if we could show specific binding of ligand to receptor. Here, we did this by setting up a radioligand binding assay. After the receptor is extracted and incorporated into our detergent micelles, the sample is incubated with radio-labeled ligand. We determined specific binding by using a competition assay of excess cold or unlabeled ligand to hot or radio-labeled ligand. And then the activity that we saw in these competition samples was considered to be our non-specific binding. Post-incubation, the free radioligand is removed via gel filtration, and then our cleaned up sample is added to vials containing scintillation fluid and read on a scintillation counter. The scintillation counter gives us our CPMs, or count per minute, and from there we calculate the DPMs, or disintegrations per minute, by dividing the CPMs by the efficiency of our radio label. In our case, we use tritium-labeled ligands. In this experiment, we are looking at the muscarinic acetylcholine receptor 3, represented here as M3. Muscarinic receptors are located in many places in the body, including the smooth muscles of blood vessels, the lungs, and in many glands, which help to stimulate secretion. In particular, M3 has been linked to type 2 diabetes as the receptor responsible for cholinergic regulation of glucose-induced insulin release. Here we are looking at M3 expressed in our gene blazer CHO-K1 cell line and extracted with and without the presence of stabilizer in our reagent. When looking at the extractions by Western blot, we can see good solubilization of the receptor with and without stabilizer, indicating that the detergent is doing all of the extractions. In fact, they look almost identical. However, when we look at the activity by binding tritium labeled 4-DAMP, a known antagonist to M3, we see that the stabilizer is essential for maintaining functional integrity of M3 and maintains that function over a 10-day storage. Whereas when the stabilizer is not present, you lose that functionality. In fact, in this graph, you can see that when compared to a fresh extract, we're able to maintain greater than 75% of the receptor functionality when storing 1 month at -20 degrees Celsius. Now that we have confirmed functionality in one receptor, we decided to go ahead and look at a few more. In this graph, we show successful extraction of 2 receptors, again expressed in our gene blazer CHO-K1 cell lines. The dark blue bar is M3, as we discussed in the previous slide, and F5-HT1A serotonin receptor 1A is the yellow bar. Serotonin receptor is a drug target for depression and anxiety. Serotonin 1A receptor is also associated with Alzheimer's and Parkinson's disease and has the potential for therapeutic treatment. Here we show serotonin receptor extracted with the GPCR extraction and stabilization reagent is specifically able to bind tritiated propanolol, a no antagonist for this receptor. Lastly, we looked at an endogenous adenosine receptor type 2A, or ADOR2A, extracted from mouse brain tissue, whose specific activity is represented in the light blue bar. Adenosine receptor 2A plays an important role in the brain and regulates the release of neurotransmitters like dopamine and glutamate, and has been found to be associated with depression, insomnia, pain, Parkinson's, and Huntington's disease. In the graph on the right, we stored the active receptor for 2 weeks at 4 degrees Celsius and found the ability to bind its agonist, adenosine, was maintained when directly comparing to a freshly extracted receptor. So we have shown that the GPCR extraction and stabilization reagent efficiently solubilizes active and stable GPCRs, but how does it compare to other techniques? In this experiment, we looked at 2 methods. The first method is ours, and here's a quick recap. First, we washed our sample. In this case, we used mouse brain tissue and resuspended in cold GPCR extraction and stabilization reagent, then doused to homogenize. We transferred our homogenate to a new tube and incubated for 30 minutes at 4C. Post-incubation, we collected cell debris with centrifugation, and in about an hour total, you can collect your lysate containing your micelle encapsulated GPCRs. The second method we tried was SMALT, which is a detergent-free solubilization of membrane proteins by styrene maleic acid copolymers, also known as SMAs. This method has been a new focus for detergent-free preparation of cryo-EM samples. First, we washed the sample and resuspended in a ml of a lipid solution. We used 10 mg/ml DMPC and homogenated with a Downstream homogenizer. The homogenate was then transferred to a new tube and incubated at 37 degrees C in a water bath sonicator for 30 minutes. Post-incubation, a ml of polymer was added dropwise to the solution, and it was allowed to equilibrate at room temperature for 1 hour. In this case, we used 2 different polymers, LipoDisc 3.1 and Amphipol. The equilibrated samples are then spun down at 40,000 times G for 30 minutes in an ultracentrifuge, and in about 3 hours total, you can collect your lysate containing GPCRs surrounded by lipid, which is bound by the polymer. In this data set, we are looking at those mouse brain extracts prepared with the 2 different methods just shown in the previous slide. The Western blot in the upper left is showing the solubilization of metabotropic glutamate receptor 3, or mGLR3, and beta-2 adrenergic receptor, or B2AR. mGLR3 is associated with mental health disorders such as bipolar affective disorder and schizophrenia. As you can see in the blot, there is more mGLUR3 present in the samples prepared with our method than with the SMALT method used for the other two samples. Solubilization of B2AR, which is known to be located in the cerebellum and is involved in brain to immune system communication, is similar across all methods. We then took these same 3 extracts and performed radioligand binding assays to look at the activity of adenosine receptor, endogenously expressed within the mouse brain. In the graph on the bottom left, the highest and most specific binding of adenosine is seen with the extract made using the GPCR extraction and stabilization reagent, indicating that we had better solubilization of functional adenosine receptor using this method. From this data, we can conclude that the GPCR extraction and stabilization reagent can successfully solubilize functional GPCRs from the cell membrane. There are multiple techniques to consider when trying to solubilize membrane proteins. An extraction using non-denaturing detergents can be just as efficient as other techniques when optimized and can even improve Western results. In order to maintain receptor function, it is critical to perform the extraction and stabilization in conjunction with each other, allowing the receptors extracted with this reagent to remain stable 1 to 2 weeks at 4 degrees Celsius and up to 1 month at -20 degrees Celsius. When looking at the solubilization of 3 different receptors, we saw an increase in receptor function when using the GPCR extraction and stabilization reagent. When compared to reference techniques using styrene maleic acid or amphipathic polymers for stabilization of adenosine receptor, extracts made with the GPCR extraction and stabilization reagent shows greatly increased function of receptor initially and also over time. Thank you for your time, and I will be happy to take any questions at the conclusion of our talk. I will now hand the ball over to Betsy to discuss the isolation of cell surface proteins.
Thank you and hello. As Joanna has shown you, extracts of whole cell lysates are a convenient and easy way to obtain GPCRs, but this method is often not sufficient for detection of low abundant membrane proteins. Sometimes it's necessary to enrich to see more. In this presentation, I will describe an improved method for the isolation of mammalian cell surface proteins. I will first provide an overview of the challenges with membrane protein isolation, followed by commonly used isolation methods. Next, I will walk you through the amine-based labeling chemistry we chose for enrichment. The enrichment protocol includes biotinylation of cell surface proteins, cell lysis, capture of labeled proteins, wash, and elution. Details on how to avoid off-target labeling will be presented along with the key steps we focused on for protocol optimization, including selection of detergent for membrane protein solubilization, as well as selection of wash buffers that allow compatibility with mass spectrometry. In addition, the breadth of enriched proteins that can be obtained will be demonstrated through a case study where we isolate the surface proteomes of 3 human breast cancer cell lines and analyze them with mass spectrometry. Efficient isolation of cell surface proteins is challenging for many reasons. Cell surface proteins are predominantly transmembrane proteins that contain 1 or more hydrophobic domains residing within the lipid bilayer. The hydrophobicity of these proteins results in poor solubility in common extraction buffers. In addition, cell surface proteins are expressed at lower levels compared with the much more abundant intracellular proteins, and they require enrichment prior to proteomic analysis. The most common isolation methods for cell surface protein isolation are shown in this table. Ultracentrifugation is the oldest method, relying on physical properties of the plasma membrane, such as size, shape, and buoyant density for separation. Unfortunately, this method requires a large amount of starting material and is plagued with low purity. Alternative chemical isolation methods have become much more popular. Typically, biotinylation labeling reagents are used due to the ease of affinity purification with biotin binding proteins, such as streptavidin. There are 2 common methods that are used. The first involves biotinylation of primary amines through NHS esters. Primary amines are plentiful on the cell surface as they are found in lysine residues. The procedure is fast and results in a high yield of enriched proteins. Unfortunately, some biotin NHS esters can also cross the plasma membrane through biotin transporters or diffusion, leading to intracellular labeling and contamination of the enriched fraction. An alternative biotinylation method involves labeling of glycoproteins on the cell surface. Here, oxidation of N-glycoproteins is performed prior to cell lysis, so only extracellular carbohydrates are modified. Following lysis, biotin hydrazide is used to label the oxidized glycoproteins. This method avoids off-target labeling, but it's lengthy, requires large starting amounts, is limited to N-glycoproteins, and results in low yield of the enriched fraction. We chose the amine-based biotinylation method because it's much faster than the other methods and has a much higher yield. Our goal was to minimize intracellular labeling, thereby enriching cell surface proteins with minimal contamination. Sulfo-NHS-SS biotin, shown here, is the labeling reagent we use to modify cell surface proteins. NHS chemistry is very simple and efficient, involving the reaction between a reactive ester, shown here in red, with a primary amine on your protein. The sulfo-NHS group renders the labeling reagent water-soluble, making it impermeable to the lipid bilayer. The biotin on the linker, shown in teal, allows for capture of the modified proteins onto a biotin-binding resin. Once bound, the disulfide bond located in the spacer arm, shown here in purple, allows for detachment of the bound proteins from the resin using a reducing agent, such as DTT or TSEP, which breaks the disulfide bond. Upon release, the protein retains a small remnant of the labeling reagent. There are several key steps to improving the selectivity of enrichment. The first is biotin labeling. Although minimizing the amount of label used and the labeling time are important, treatment of unreacted label post-labeling is critical. Most methods involve quenching unreacted labels, but we found that the quenching reagents are not nearly as effective at reducing intracellular labeling as simply removing the label and washing the cells. This Western blot shows the difference between the two methods. Enrichment of HeLa cell surface proteins was performed in duplicate with the improved method using label removal. A common amine-based method from the literature that uses a quench buffer was used as a control. The latter is referred to as the original method. The lanes with the eluates are outlined in teal for easy identification of the enriched fractions. Focusing on the bottom of the blot, F stands for flow-through, E for elution, and B for post-elution bead boil. Common intracellular contaminants, actin and HSP90, were found to heavily contaminate the elution fractions with the original method, but almost negligible amounts of these proteins were seen in the eluates with the improved method. One of the top complaints with biotin labeling amine groups is that the label can cross the cell membrane and biotinylate intracellular proteins. This next slide shows the location of the biotin label on the cells using our improved protocol. MCF7 cells were fixed, permeabilized, and then stained with NeutrAvidin 488 conjugate, a green stain. The nuclei were counterstained with Huxt, a blue stain, and the cells were imaged on a fluorescent microscope at 63x magnification. The images on the left were prepared with cells that were labeled with biotin. Focusing on the upper left quadrant, which merges both stains, the cells clearly show label on the surface only. In contrast, the images on the right are control cells that were prepared without biotin label. As expected, these cells do not show staining with NeutrAvidin 488. A second key step for optimal enrichment is the choice of detergent for cell lysis. The challenge is to find a balance between solubilizing all proteins and selectively extracting just what you want. We chose a detergent that solubilizes membrane proteins better than the commonly used detergents in the literature. The Western blot shows cell surface proteins isolated from HeLa cells using the two different detergents. Similar yields with both detergents was seen with EGFR and cadherin, both single spanners. In contrast, higher yields of multi-spanning membrane proteins, sodium potassium ATPase and thrombin receptor, were seen with the improved detergent. Choice of affinity resin is also critical for enrichment. We compared NeutrAvidin and streptavidin resins and were surprised to discover that NeutrAvidin performed better. Biotinylated cell surface proteins were captured on both resins and then eluted at room temperature. As seen in the Western blot, protein yield in the elution fractions was much higher with NeutrAvidin. We hypothesized that the binding pocket for streptavidin is deeper than that for NeutrAvidin, potentially shielding the disulfide bond from exposure to reducing agents and making it harder for release of labeled protein under room temperature elution conditions. Interestingly, we have seen similar performance for the resins when elutions were performed at 95 degrees. The combined results of the improved protocol are shown here. Western blot analysis was performed on enriched fractions from two different cell lines, HeLa and HCT116, that were processed using the isolation procedure. Samples were run in duplicate and normalized by volume. The data show high yield of 4 cell surface protein targets, seen on the top of the blot, with negligible contamination of 4 intracellular proteins, seen on the bottom of the blot. Our optimized protocol for cell surface protein isolation is summarized in this schematic. Adherent cells are first labeled with sulfo-NHS-SS biotin for 10 minutes at room temperature. The label is then removed and the cells are washed twice. This step is critical as any unreacted label can increase chances of intracellular labeling during cell harvest. Labeled cells are then gently scraped off the plate into buffer and centrifuged. The cell pellet is lysed for 30 minutes using a detergent that efficiently solubilizes multi-spanning membrane proteins. Labeled proteins in clarified lysate are then captured on NeutrAvidin resin for 30 minutes. The resin is subsequently washed with a stringent mass spec compatible wash buffer, and the captured cell surface proteins are eluted off the resin using 10 millimolar DTT for 30 minutes. Isolated cell surface proteins can then be analyzed by Western blotting or by mass spectrometry. This protocol can also be used with suspension cells using 1 to 2 times 10 to the 7th cells. Up to this point, I've used Western blotting to demonstrate cell surface protein enrichment. Another common method to look at isolated cell surface proteins is mass spectrometry. There are a few key requirements that we address to ensure compatibility of our cell surface protein isolation protocol with mass spec. Although detergent is used for solubilization of membrane proteins, it's not compatible with mass spec, so it had to be removed. Although salts are important in wash buffers for the removal of contaminants, they are also incompatible with mass spec and had to be removed. Lastly, elution conditions had to be modified to allow for compatibility with MS sample prep. As a side note, and unexpectedly, we found that DTT was significantly more efficient at eluting biotinylated proteins versus TSEP. To perform mass spectrometry analysis on a sample, the washed resin prepared in the cell surface protein isolation method was processed with a slight modification to make it mass spec compatible. After washing the resin with a wash buffer that contains detergent and salt to remove the nonspecific binding, the resin is washed 3 times more with triethyl ammonium bicarbonate to remove the excess detergent and salt. Following elution with 10 millimolar DTT, the Thermo Scientific EasyPep Mini MS sample prep kit was used to prepare the peptides for mass spec analysis. The peptides were analyzed by LCMS using a Thermo Scientific QE Plus mass spectrometer. The raw data was searched using Thermo Scientific Proteome Discoverer software. The minimum criteria for protein identification was 2 peptides with a high confidence based on the software algorithm. Plasma membrane proteins were identified using the UniProt knowledge base. There are different ways to quantify mass spec data. Label-free quantitation is the simplest method and involves using the MS spectra peak intensities to estimate protein relative abundance. The label-free method was used in the next series of data I will share with you. An alternative method uses isobaric tags. Isobaric tags, such as the Thermo Scientific Tandem Mass Tags, or TMT reagents, are chemical groups with identical masses that vary based on the distribution of heavy isotopes around their structures. This method allows for increased throughput and improved quantitative precision. I will discuss these tags in more depth later in the presentation. In this next slide, we used label-free quantitation to demonstrate the effectiveness of our improved method. This figure shows the number of cell surface proteins and intracellular proteins identified by mass spec in the top 100 most abundant proteins isolated from A549 cells. The improved method is compared with an original literature method and a whole cell lysate. As you can see, the improved method yielded 5 times more cell surface proteins than the original method, with significantly fewer contaminants. In fact, the original method was found to be only marginally better than a whole cell lysate. The same data set in the previous graph was used to compare the abundance of selected cell surface proteins across the improved method, original method, and whole cell lysate. Samples were prepared in duplicate and the data was normalized by peptide yield. 2 sets of 10 proteins each, spanning 2 orders of magnitude in abundance, were compared. The data demonstrates significantly higher abundance of cell surface proteins with the improved method. It's important to note that at the lower abundance levels, that is, at a magnitude of 10 to the 8th, the data set for the original method did not contain 3 of the 10 proteins that were seen with the improved method. In this next experiment, cell surface proteins were enriched from 3 cell lines, including HCT116, A549, and HeLa. Following mass spec sample preparation, the peptide yield was determined and found to be similar across all 3 cell lines, measuring 10 to 14 micrograms of isolated peptide per sample, as shown in panel A. Following MS analysis, the top 50 most abundant proteins were manually reviewed in the UniProt knowledge base. Panel B shows the classification and number of cell surface proteins, including single-spanning or multi-spanning transmembrane proteins, the latter with spans ranging from 6 to 17 spans, glycosylphosphatidylinositol or GPI-anchored proteins, and extracellular matrix proteins. A similar distribution was seen across all 3 cell lines. Examples of these different types of enriched cell surface proteins are highlighted in the lipid bilayer shown on the right. These initial label-free mass spec studies provide confirmation that our improved isolation method identifies more proteins in higher abundance than the original method. To further validate our new protocol at a deeper, more quantitative level, we performed a more relevant case study using mass spec quantitation with isobaric labeling. The case study was set up to evaluate differences in the surface proteomes of 3 breast cancer cell lines. The cell lines included MCF7, which is responsive to both the estrogen receptor alpha and progesterone receptor, and also 2 triple negative cell lines were evaluated, BT549 and HS578T. These are negative for the estrogen and progesterone receptors as well as HER2/neu amplification. The study was performed using chemical tagging for the highest quality identification of differences between these unstimulated cell lines. Where possible, results were confirmed with the literature. TMT or isobaric labeling tandem mass tags were used for multiplexed protein identification and quantitative analysis by tandem MS. A TMT tag consists of an amine reactive NHS ester group, a spacer arm, and a mass reporter. 9 different TMT tags were used to label triplicate samples from each cell line as shown. The labeled peptides were then combined and fractionated with high pH reversed phase columns. Fractionation increases the number of peptide identifications due to reduced sample complexity. For each sample, a unique reporter mass in the low mass region of the MS-MS spectrum is used to measure relative protein expression levels during peptide fragmentation and tandem MS. In the case study performed here, the data is expressed relative to the HS578T cell line, which is assigned a value of 100%. Shown here is a heat map of the enriched proteins in the 3 breast cancer cell lines. The surface proteomes for the 3 unstimulated cell lines are clearly distinct based on the clustering of protein groups. Minimal variation is seen across triplicate samples for each cell line. Based on this data, we pulled out interesting proteins from 3 different signaling pathways to compare and contrast across the 3 cell lines. The first class of enriched cell surface proteins we evaluated was tyrosine kinases. Focusing first on the mass spec data at the bottom of the slide, the epidermal growth factor receptor, or EGFR, was found to be more highly expressed in the triple-negative breast cancer cell lines, from here on referred to as TNBC, compared with the hormone-responsive MCF7 cells. EGFR is a major regulator of cell proliferation and cell survival. In TNBC patients, EGFR overexpression is often observed, consistent with the BT549 and HS578T data shown here, and is associated with poor clinical outcomes. Anti-EGFR drug therapies have been unsuccessful with TNBC patients, but recent studies have gained traction with combinatorial therapies. Switching to the receptor tyrosine protein kinase ERBB3, significantly higher levels of this receptor were found in the MCF7 cells compared with the TNBC cells. The ERBB3 receptor, also known as HER3, is emerging as an important molecule in estrogen receptor positive breast cancers. Levels of this receptor have been shown to be very low in BT549 and HS578T cell lines in the literature, consistent with the data shown here. This mass spec data for EGFR and ERBB3 correlates nicely with the Western blot data of these same targets shown at the top of the slide. It's important to note the improved sensitivity and better quantitation with the TMT mass spec data compared with the Western blot data, due to the fact that the MS data are not antibody limited. The next class of proteins we evaluated was GPCRs. Several GPCRs were identified in our case study, and variable levels were found across the 3 breast cancer cell lines. CD97 is an adhesion GPCR involved in tumor cell migration and invasion. It was the first adhesion GPCR to be linked to cancer. Adhesion G-protein coupled receptor L2, as the name suggests, is also an adhesion GPCR. Both have been associated with TNBC. Frizzled receptors are a family of GPCRs that bind to secreted wingless type, or Wnt, ligands in the Wnt signaling pathway. Frizzled 7 plays a role in cancer development and progression and is an emerging target for cancer therapy. It is most commonly upregulated in a variety of cancers, including TNBC. The final class of enriched surface proteins we looked at was cell adhesion proteins. Cell adhesion molecules play a key role in metastasis by regulating both cell-to-cell and cell-to-matrix interactions. Cell surface glycoprotein MUC18 messenger RNA expression has been shown in the literature to be high for the BT549 and HS578T cell lines, but low for MCF7, in agreement with the mass spec data shown here. Neural cell adhesion molecule 1 has been shown to be highly expressed in BT549 cells, but absent in MCF7 cells, also in agreement with the data shown here. Lastly, neural cell adhesion molecule L1 has been shown in the literature to promote migration of MCF7 cells. To sum up, this case study uncovered significant differences in proteins across hormone-responsive and triple-negative breast cancer cell lines. It's important to note that the proteins discussed are only a very small fraction of the total number of proteins identified using TMT mass spec analysis. In fact, this combination of enrichment with quantitative mass spec revealed a treasure trove of proteins that just 5 years ago could not be identified. In particular, I found a 2014 paper where the authors evaluated the plasma membrane proteomes of several breast cancer cell lines and could only identify one GPCR, which is CD97. Today, we have superior methods and more sensitive instruments, allowing for a gold mine of biological information that just a few years ago was unattainable. In order to obtain the data I've shared with you today, several different Thermo Fisher Scientific kits were used. They include the Pierce Cell Surface Protein Biotinylation and Isolation Kit, the EasyPep Mini MS Sample Prep Kit, the TMT 10-plex Isobaric Mass Tagging Kit, and lastly, the Pierce High pH Reverse Phase Peptide Fractionation Kit. Combined, these kits create a workflow for selective cell surface protein enrichment, cleanup, and high sample throughput with quantitative precision. In conclusion, I have demonstrated the following: an improved method for isolation of cell surface proteins using amine biotinylation results in significantly fewer intracellular contaminants and allows for better selectivity and enrichment of cell surface proteins. The improved method and reagents for cell surface protein isolation are compatible with mass spectrometry. Isolated cell surface proteins include a broad range of membrane proteins, including single and multi-spanning transmembrane proteins and GPI-anchored proteins. And lastly, isolated surfaceome content can be used for identification of potential drug therapy targets, including GPCRs. In future studies, we plan to stimulate the cells with growth hormones to detect changes in cell signaling and the surfaceome content. This concludes our presentation. Thank you for your attention. Joanna and I are happy to answer your questions.